It began a long time ago, in our own Milky Way galaxy, during a hangout hosted by Dreadnought Silvers. TrueEmpiricism had made a claim that would forever change the great debate community. A claim that violated everything any of us knew, or thought we knew, about chemistry, biology and perhaps even our very reality as a whole. TrueEmpiricism claimed that when DNA was dissolved in water, it meant the same as DNA degrading in water. He further claimed that if DNA was put into water it would lose its nucleotide sequence and break apart into its constituent parts. Well, what did he mean by this? No one really knows for certain. However, this video will show exactly what does happen when DNA goes into solution, and then compare that to the many things TrueEmpiricism has said since his initial discussion with Dreadnought. But first, let's go back to the very beginning, back to that fateful day, and to the discussion that started it all.
The fact that you can dissolve one into the other does not mean that it's going to compromise its structure.
What do you mean dissolve? What do you mean dissolve one into the other?
So if you have, when you dissolve something, doesn't mean that it actually it loses its how can I say this? Its integrity. So if I have cooking oil and I pour cooking oil into water, I can actually strain out that cooking oil and reuse it. It doesn't just become unusable. Does that make sense?
Well, actually, I would disagree with that wholeheartedly. If you're having DNA,
they're admissible.
If you're having DNA breaking down to its more constituent parts, and you're going to say that you can reuse that, I don't know a system that at all is totally run on only the chemicals and the broken down parts of DNA.
Dreadnought was describing a solution to TrueEmpiricism, which is comprised of a solvent and a solute, evenly mixed together. In this example, sugar, which is the solute, is being dissolved into the water, which is the solvent. The sugar molecules remain sugar molecules, and do not break apart, they remain sugar. The only thing that breaks apart, as shown here, is the lump of sugar, which by diffusion randomly and equally distributes the sugar throughout the solution, but the sugar itself does not degrade, disintegrate, nor are any molecular bonds broken. And if the water was removed, the sugar would be a pile of sugar, which we could once again make into a lump just as it was before, as if it going into solution never even happened.
Okay, so since we're dealing with a broken down system, all right, well, what system do we know of that runs on broken down parts of DNA and not a strand that has sequences of DNA in it?
Okay, and when you're saying broken down, does that mean that there is a chemical reaction, yes or no?
Didn't I just answer you that on that a few minutes ago?
I'm trying to go through it because every time I continue with my line of thought, you interject, and I'm trying to pin down what this reaction is. I mean, it's either a gas forming reaction, a combustion reaction, an exchange reaction, an acid base reaction. It would be a base
reaction. You can look at it would be a base reaction, I believe.
A base reaction.
You could probably look at it as I forget the word for it. Decomposition reaction.
A decomposition reaction, which means to break apart, is the opposite of a synthesis reaction, which means to join together. A decomposition reaction occurs when a larger molecule breaks into smaller sized molecules. Two water molecules, undergoing electrolysis, where energy is going into the system to break the molecular bonds, would undergo a decomposition reaction to form two diatomic hydrogen molecules and one diatomic oxygen molecule. But when sugar or DNA go into solution, no structural molecular bonds are broken, no smaller molecules are being produced, and therefore no decomposition reactions are taking place. I think now is a good time to describe what actually happens when DNA goes into solution. Here we have a strand of DNA showing its constituent parts. It is a double helix with a sugar phosphate backbone, with joining complementary nucleotides in the middle. Each nucleotide being a monomer, or the smallest unit of DNA, and are comprised of a nitrogenous base, which are a T, C or G, a deoxyribose sugar, and a phosphate group. The phosphate group normally is a phosphate atom with a double-bonded oxygen atom, an oxygen atom with phosphodiester bonds to the sugar and base, the sugar and base being known as a nucleoside, and OH groups that are electrically neutral. When DNA goes into solution, since it being an acid, will donate the hydrogen atom to the water, called protonation, which will eventually combine with the water molecules, or H2O, to form a hydronium or H3O. Now let's rewind time again, and see what happened when molecular biologist Dr. Fiona Robertson attempted to correct true empiricism by trying to explain to him that DNA dissolves in water, which remember means to go into solution, and DNA will only have a positive H ion come off the OH from the phosphate group, but structurally stays perfectly intact, and does not degrade or break down into its constituent parts.
How are you going to sit over here and say that DNA is water soluble, but you put it in DNA or in a pure water and it stayed intact? That's a contradiction.
It stayed intact in solution.
The contradictory dictates that didn't happen.
No, Ronnie. Ronnie, no. I'm telling you, I've done it. I dissolved the DNA in ultra pure water. The DNA remains intact.
It stays.
You can check it on an agarose gel.
You're saying that you've done it, but we can't confirm that here. I don't know.
You can actually. It's published.
I know it's in the textbooks, and I know what the
No. Go and look at my publications. Go and look at my research publications. The full methods for the isolation and the cloning and the sequencing and the manufacture of the radioactive probes is all the full method.
How about this?
It's published.
How about you show me a publication to where you've done DNA in pure water, and it says that pure water with nothing added into the solution. And I can see that you're
Ronnie, are you actually trying to suggest that I haven't?
Live right here in front of everybody.
True Empiricism had falsely accused Dr. Robertson of lying when she said that she had dissolved DNA in water and it stayed intact. Anyone who has ever worked or studied in a biology lab has probably done a polymerase chain reaction, known as a PCR reaction. This is where small amounts of DNA are amplified to produce millions of copies of a DNA strand or fragment. True empiricism was incredibly in error and was being corrected by a PhD who is an expert on this topic, but he continued to claim that Dr. Robertson was wrong. So Hogtie Champ and him sat down to try to suss out what he meant when he says that DNA would break apart into its constituent parts when it dissolved into solution.
The problem is that where the polyanion is on the DNA, that's on the sugar phosphate backbone. So the sugar phosphate backbone, if you screw around with that, that's going to render it unstable. So I think that's the difference here. Where the polyanion is on the DNA, that is what is going to affect the whole strand, both strands of DNA. So I think that would be the difference between what you're saying.
Okay, so you're saying I'm going to agree with you there that since the portion that I'm talking about, the nut phosphate group, if it were the case that you messed with that connection, maybe that release of the proton, that is, it's an acid, it's a proton donor, maybe that would be the example of like how train cars are very tightly coupled together, but all you have to do is pull a pin out. Actually, that's not true. People haven't used pins for years. Tractors, they use the pin. There's a tongue and then the thing like this and a pin. Is that no matter how tight that thing is, all you have to do is bink, pull the pin out, and now it disconnects, it uncouples. And is it the case that this proton coming off in solution is the thing that would break apart the phosphodiester bond and liberate whole nucleotides? Is that what you're suggesting?
Yeah, well, that's pretty much what I'm suggesting. If this is dependent on the stability of the strand, and that is what's screwed up, then it's going to be unstable. So if you affect the polyanion in DNA, then that's basically what's going to happen.
This is where true empiricism shows his confusion as to what actually happens when DNA goes into solution. The linchpin analogy that Hog Tai Champ was using would have been more in regards to the oxygen atom with the phosphodiester bonds on the sugar phosphate backbone, here shown in a red circle. However, true empiricism did not realize that the H ion being produced is not from that oxygen atom, but from the OH group on the phosphate, circled in green.
Yeah, well then at that point, that's where I wouldn't agree with you, because this phosphate group, it's a terminal group, right? Not the phosphate, the hydroxyl group on the phosphate segment of the nucleotide is a terminal thing. So all it is you've got a little bit of a negative charge around that one oxygen that's sort of off to the side, and then, well, the proton leaves without taking its own electron with it, so it leaves that as a negative charge at that oxygen. But the connections back up here, and there's other covalent bonds that are back there. So just the liberation of that proton, it was already an imbalanced charge. There's that polar property there on the hydroxyl group, and all it is it just becomes slightly more polar.
Hog Tai Champ correctly notes that the OH is a terminal group and is not part of the linkage of the sugar phosphate backbone, nor has it anything to do with any bonds between the nucleotides. The loss of the hydrogen ion to the surrounding water would have absolutely no effect on the structural stability of the DNA molecule while it is in solution. Remember, the smallest component or constituent part, called a monomer, of DNA is a nucleotide. True empiricism told Hog Tai Champ that DNA would be breaking down into segments, and the segments of DNA are the nucleotides. Again, this does not happen when DNA goes into solution, as the only thing that happens is the loss of a hydrogen ion, off of a terminal OH on the phosphate group. 6 months later, True Empiricism attempts to explain a little more in detail by what he means by claiming that dissolving means that DNA breaks down in water, only making his position even more confusing to everyone in the community.
Let's get a more detailed description of what is actually happening right here with dissolution. Dissolution is the process, and I hope you guys can see what I'm screen sharing, you guys. Dissolution is the process through which a solute forms a solution and a solvent. The solutes, and of course we just got a pretty good description just now from this lady. The solute is the case of solids, has its crystalline structure disintegrated as separate ions, atoms, and molecules form. Well, what did that just say right there? The solute, in the case of solids, has its crystalline structure disintegrated as separate ions. What does disintegrated mean? Disintegrated means something that breaks up into small parts. Something that breaks up into small parts. Now, we know that this is exactly what happens right now when something is soluble. The process that undergoes something that is soluble, it breaks up into parts. Already now, from the very first video that the first individual showed that they were playing stupid on, it confirmed exactly what I was saying word for word. That DNA, for example, what I've been saying from the beginning, DNA will stay DNA, but will break down into specific sequences. They compared that with the covalence compound of sugar, staying sugar, but breaking down.
They compared DNA to sugar because, like sugar, DNA consists of covalent bonds, not ionic bonds, which true empiricism had been describing. Ionic meaning that there is a complete transfer of electrons, resulting in the sodium becoming negative when it donates an electron to the chloride, which in turn takes on a negative charge. When salt enters solution, the crystals of salt dissociate into sodium and chloride ions. This happens since water is a polar solvent. It can therefore break apart polar bonds. DNA does not do this when it goes into solution, as it is a polar molecule, but with non-polar covalent bonds, not ionic. Covalent bonds are where two atoms share two electrons rather than a transferring of an electron, and would require a covalent solvent to break the DNA apart, which water is not.
And they pretty much ignored where it said breakdown and only paid attention to the fact that it said that it was going to stay sugar. Remember that DNA, like as I said, that it has to be in specific sequences. It will no longer be in specific sequences afterwards. To break down or to deteriorate mean what does it mean then when something is dissolved? What is the process of dissolution? And everything means that it's going to disintegrate, break down into smaller pieces, and that's what it means. And this is synonymous with saying that it is to degrade.
Synonyms are words that can generally be used interchangeably as they colloquially mean the same thing. However, in science, words have specific meanings and are not always completely interchangeable, as they often imply or infer different things. Please note this carefully, as true empiricism will later on make the complete opposite claim, saying that dissolve and degrade are in fact not synonyms, as everyone had been trying to tell him, and in the context of chemistry do indeed mean completely different things.
I'm screen sharing here. When I think that the DNA strand is going to break down, I think that it's going to stay. Independent monomers, they're going to stay attached to each other, right? You're going to have a whole section of deoxyribonucleic acid, the sugar phosphate sections completely attached. What I'm thinking is going to happen is that it's going to break down through the middle, because it's going to be pulled apart from both ends by these negative charges, and more than likely the adenines and the thymines are going to probably be coming apart, because the adenines and the thymines, their strength, I think their strength holding together is weaker than the other segments.
True empiricism here has now moved away from his claim that the phosphodiester bond, the bond of the sugar phosphate backbone, to claiming that the hydrogen bonds in the middle of the DNA molecule would break apart when DNA goes into solution. This is bullocks, as the center region of DNA is a hydrophobic region, and the hydrogen bonds breaking is a function of water temperature known as a melting curve. The hotter the water, the more hydrogen bonds would break. But the discussion has been what happens to DNA when you place it into an aqueous solution at room temperature, which wouldn't have sufficient energy to break any of the hydrogen bonds.
Again, I think that we've accomplished a lot with this. We've proven that DNA is, when something is soluble, that means it's going to break down. That doesn't mean I'm not saying that the individual atoms are going to come apart. When I say it's going to break down to its constituent parts, I'm saying that it's going to break down to the individual sections that make it the DNA, deoxyribonucleic acid.
True empiricism often refers to DNA as deoxyribonucleic acid, but deoxyribose is the sugar in the DNA molecule, with the molecule itself being called deoxyribonucleic acid. It is called deoxyribose due to the missing oxygen atom on the 2' part of the carbon ring.
But what I'm saying is that that doesn't necessarily mean that it's going to break down all the way to the atoms. It's just the independent sections of the molecule that make it deoxyribonucleic acid. I mean, obviously, breaking the covalent bonds, like right here, like that would probably have to undergo, some sort of hydrolysis reactions.
Hydrolysis is when a bond is cleaved or broken by water. Water, again, being a polar solvent, would not break the covalent bonds in the DNA, and neither the hydrogen bonds nor the glycosidic bond between the sugar and base would undergo hydrolysis. It is the opposite of a condensation reaction, where a bond is formed and water is a byproduct of the reaction.
If I'm saying that DNA is going to stay DNA, but it's going to break down to its constituent parts, and you guys keep on saying, well, that doesn't mean it's going to degrade. It doesn't mean it's going to break down. All it means is it's going to go into solution, and the descriptions all the way across the board are saying it's going to break down. It's going to disintegrate, which means it's going to break down. degrade means, chemically speaking, that it's going to deteriorate or break down chemically. Well, then what is the problem here? I mean, because like really, literally, word for word, all the way across the board, I'm 100% right. There's not the slightest implications in any way at all that I'm wrong.
Quite the contrary, as everything points to the case that true empiricism is wrong across the board when he is claiming that DNA degrades in water, and that dissolve, degrade, and disintegrate all mean the same thing, contrary to everyone, as he notes, who had been consistently trying to explain to him that they are not synonymous terms.
Keep in mind that this is not what I'm talking about at all. I have not been talking about an hydrolysis reaction at all. I've been talking about dissolving. That's what I've been talking about. I've been talking about that sugar phosphate backbone, and because it has a partial negative charge at a cellular pH, that once it starts to attract those water molecules, it's going to start to break down because of that. Now, that doesn't mean that it's going to start breaking apart into little individual pieces. That means that it's going to stay DNA, but it's not going to be in sequences anymore. It's not going to be some long strand of DNA that's going to be, in specific sequences. And in life, you need DNA in specific sequences because that's what's actually important. So I guess what I'm saying here is that we're not only having to deal with the situation of DNA breaking down in water, and I've shown articles and different sources, like I showed the other day, talking about how DNA is really fragile and will break down. Her video actually showed that too, that DNA will break down in water. It just won't break the same way that ionic compounds will break down. So I've confirmed this already, but this isn't the only problem now. But we're also now having to deal with what she's been talking about with hydrolysis reactions. We have to deal with the individual guanines and thymines and cytosines breaking down.
True empiricism here is claiming that the covalent bonds of the individual bases, known as nitrogenous bases, will break down in water. However, water being a polar solvent, it would not break the covalent bonds of the individual bases. Let's recap true empiricism's claims. DNA will degrade in water. Dissolve, degrade, and disintegrate are scientifically interchangeable. DNA will break down into its constituent parts. The phosphodiester bonds of the sugar phosphate backbone will break. The hydrogen bonds between the bases in the middle of the DNA molecule will break. The covalent bonds of the individual nitrogenous bases will break. But what really happens when DNA goes into solution is only a single hydrogen ion comes off an OH terminal from the phosphate group.
The scientific phenomena that happens when DNA breaks down in water versus that of dissolving. Dissolving and degrading is different, scientifically speaking.
Which is exactly what everyone had been trying to tell true empiricism since the very beginning. Has the penny finally dropped for him? I sincerely doubt it, as he is so incredibly inept at biology that I'm certain he will just keep making the same puerile mistakes over and over, as he is forever lost in any subject relating to science.
But I'm right. I think you guys just need to acknowledge that already.